Which of the following enzymes is crucial for the process of polymerase chain reaction, PCR, to amplify DNA? A. Reverse transcriptase. B. DNA ligase. C. Taq polymerase. D. RNA polymerase. The correct answer is C. Taq polymerase is a heat-stable DNA polymerase used in PCR to replicate DNA during cycles of heating and cooling. Its stability at high temperatures allows for the denaturation step in PCR. Which of the following vectors is most commonly used for gene transfer into plant cells? A. Retrovirus. B. Adenovirus. C. T plasmid. D. Lambda-phage. The correct answer is C. The T plasmid from Agrobacterium tumefaciens is frequently used as a vector for introducing foreign genes into plant cells, enabling the transformation of plants. In CRISPR-Cas9 gene editing, which molecule is responsible for guiding Cas9 to the specific DNA sequence for cleavage? A. Guide RNA. B. DNA probe. C. Cerner. D. Transcription factor. The correct answer is A. The guide RNA, gRNA, in the CRISPR-Cas9 system directs the Cas9 endonuclease to the specific target DNA sequence, where Cas9 cleaves the DNA, allowing for gene editing. Which technique is used to separate DNA fragments based on size during genetic analysis? A. Centrifugation. B. Western blotting. C. Gel electrophoresis. D. Mass spectrometry. The correct answer is C. Gel electrophoresis is used to separate DNA fragments by size. DNA fragments are loaded into a gel matrix, and an electric current pulls smaller fragments further through the gel than larger ones. Which of the following is an advantage of using transgenic animals in biotechnology? A. Reduced ethical concerns. B. Faster generation time. C. Production of recombinant proteins. D. Increased genetic diversity. The correct answer is C. Transgenic animals are used to produce recombinant proteins, such as insulin or antibodies, making them valuable in pharmaceutical and medical biotechnology applications. Which of the following techniques can be used for the direct delivery of genes into mammalian cells? A. Electroporation. B. Northern blotting. C. Southern blotting. D. Sanger sequencing. The correct answer is A. Electroporation is a technique that uses an electrical field to increase the permeability of the cell membrane, allowing foreign DNA to enter mammalian cells. Which enzyme is used to create sticky ends during DNA cloning to facilitate the ligation of DNA fragments? A. DNA polymerase. B. Restriction endonucleases. C. Topoisomerase. D. Reverse transcriptase. The correct answer is B. Restriction endonucleases cut DNA at specific sequences to create sticky ends, which help in the ligation process by promoting the binding of complementary DNA fragments during cloning. Which of the following best describes the role of RNA interference, RNAi, in gene regulation? A. It enhances transcription of specific genes. B. It degrades mRNA to prevent translation. C. It stabilizes mRNA to increase protein production. D. It edits DNA sequences in the genome. The correct answer is B. RNA interference, RNAi, works by degrading target mRNA molecules, preventing their translation into proteins and thus regulating gene expression. Which type of cloning technique is used to generate an organism that is genetically identical to its parent? A. Gene cloning. B. Reproductive cloning. C. Therapeutic cloning. D. Somatic cell hybridization. The correct answer is B. Reproductive cloning involves the process of generating an organism that is genetically identical to its parent, as seen in the creation of cloned animals like Dolly the sheep. Which method is used to introduce foreign DNA into bacterial cells for cloning purposes? A. Microinjection. B. Transformation. C. Somatic hybridization. D. Electrophoresis. The correct answer is B. Transformation is the process by which foreign DNA is introduced into bacterial cells, often involving plasmid DNA, enabling the bacteria to express foreign genes for cloning or protein production. In the context of genetic engineering, what is the purpose of a selectable marker? A. To increase the expression of the inserted gene. B. To identify cells that have successfully incorporated the foreign DNA. C. To promote cell division. D. To degrade foreign DNA. The correct answer is B. A selectable marker, such as an antibiotic resistance gene, allows researchers to identify and select cells that have successfully incorporated foreign DNA, as only transformed cells will survive under selective conditions. Which of the following is a common application of microarray technology in biotechnology? A. Gene expression profiling. B. DNA sequencing. C. Protein purification. D. CRISPR gene editing. The correct answer is A. Microarray technology is used for gene expression profiling, allowing the simultaneous measurement of the expression levels of thousands of genes within a sample. Which of the following describes a key difference between traditional breeding and genetic engineering? A. Genetic engineering relies solely on naturally occurring mutations. B. Genetic engineering allows for the direct transfer of specific genes. C. Traditional breeding always results in faster trait improvement. D. Traditional breeding cannot alter traits related to metabolism. The correct answer is B. Genetic engineering allows for the direct transfer and modification of specific genes, which is more precise than traditional breeding, where genes are shuffled randomly. Which type of stem cells have the potential to differentiate into all cell types, including placental cells? A. Multipotent stem cells. B. Unipotent stem cells. C. Pluripotent stem cells. D. Totipotent stem cells. The correct answer is D. Totipotent stem cells have the ability to differentiate into all cell types, including embryonic and extra-embryonic tissues, such as placental cells. Which of the following is a method used to silence a gene by preventing its mRNA from being translated into protein? A. CRISPR-Cas9. B. Gene knockout. C. RNA interference. NA. D. Site-directed mutagenesis. The correct answer is C. RNA interference, NA, is a process that silences genes by degrading their mRNA, preventing the translation of that mRNA into a functional protein. What is the role of a plasmid in bacterial genetic engineering? A. It acts as a vector to introduce foreign DNA into bacteria. B. It degrades bacterial proteins. C. It catalyzes protein synthesis. D. It encodes antibiotic resistance genes. The correct answer is A. Plasmids are circular DNA molecules used as vectors in bacterial genetic engineering to introduce and replicate foreign genes within bacteria. Which method is commonly used to determine the sequence of nucleotides in a strand of DNA? A. Polymerase chain reaction. B. Sanger sequencing. C. Northern blotting. D. Southern blotting. The correct answer is B. Sanger sequencing is a widely used method for determining the sequence of nucleotides in DNA, based on selective incorporation of chain-terminating nucleotides during DNA replication. Which of the following is an example of a transgenic crop? A. Drought-resistant maize. B. A cross-pollinated wheat variety. C. Bacterial plasmid. D. Polyploid rice. The correct answer is A. A transgenic crop, like drought-resistant maize, has been genetically modified to include genes from another organism, allowing it to exhibit beneficial traits such as resistance to drought. Which of the following best describes a knockout organism in biotechnology? A. An organism with a foreign gene inserted into its genome. B. An organism in which a specific gene has been deleted or inactivated. C. An organism that expresses a gene from another species. D. An organism with mutations in multiple genes. The correct answer is B. A knockout organism has had a specific gene inactivated or deleted, allowing researchers to study the function of that gene by observing the effects of its absence. Which technique is used to detect specific DNA sequences within a mixture of DNA? A. Northern blotting. B. Western blotting. C. ELISA. D. Southern blotting. The correct answer is D. Southern blotting is a technique used to detect specific DNA sequences in a mixture of DNA, involving the transfer of DNA to a membrane and hybridization with a labeled probe.